not going to be teaching about telomeres. That's what that has to do with. So if you guys had the opportunity, would you guys live forever?
Yes.
Yeah? Was that
Sounds interesting.
interesting? Yeah. Pretty interesting. Alright, so what do you guys know about DNA replication? Somebody missed that.
Requires enzymes,
Enzymes such as?
such as DNA replicase.
Good stuff. What else do you need? Helicase? What does that do?
It's the one that means it's, right?
Yeah.
DNA
Ligase.
ligase.
Ligase. Good, good. And what does ligase do?
It's stitches together the
Good stuff. Alright. So yeah, DNA is only synthesized 5' to 3'. And since it runs anti-parallel, of course it's going to go consistently one way and then interrupted. I'll show you a picture. You guys know. So the topoisomerase unwinds the strand. The helicase breaks the hydrogen bond between the bases to unzip it. The single-stranded binding proteins attach to each strand to keep it from annealing to itself. Primase creates primers and places them so that the Telomeres can come along and replicate the strand. And the leading strand, like I was saying, is replicated continuously. The lagging strand is discontinuous. So, I already said that. Oh no, RNA primers, that's kind of important for this whole thing. Once it's done, once it's replicated, then it goes through, an enzyme is released that eats away all the primers, and then another one comes through that replaces them with DNA instead of RNA. Ligase enzyme connects all the lagging strand segments to opposite fragments. And this is a diagram, makes it a little bit easier. A little bit out of order. This is the next one you can find. Topoisomerase comes along and unwinds it. You got helicase that comes through and opens it up. The binding proteins keep it from annealing. Primase comes along and drops primers in so that the polymerase can come through and replicate it up along. And then the ligase comes through and connects all the backbone. Alright, so what are telomeres? The repeating sequence of nucleotides, the TTAGGG sequence, or opposite the other strand, holds no information. It's just a buffer that protects things like chromosomes from being eaten away, which I'll talk about in a minute. And over time they shorten, which is what causes cellular aging, and eventually the cell is shut down, and that's why when we get old, it takes longer for cuts to heal and whatnot, because cells aren't replicating anymore and producing new tissue. So the whole thing about telomeres is the end replication problem. This is the reason that they shorten. To replace RNA primers, the enzymes must have DNA on both sides of it. So when it goes along, it reads the 3' hydroxyl group, and it knows that there's a gap there, it fills it in. But on the ends of the chromosome, we just have a gap with no 3' hydroxyl on the other end to let it know that it needs to be filled in. So once the RNA primer gets dissolved, then that complementary sequence is lost. Pretty small. So pretty much, you got the regular DNA here, it gets split apart into this 2 strands. This is replicated continuously, discontinuously here. So as it goes along, it reads the 3' hydroxyl group right there. Gets rid of the primer and then puts DNA there. But then you come down here, and as it comes along, there's no 3' hydroxyl end right here. The group on the end right here. So it doesn't know to replace the DNA. So this just gets left open, and then all these base pairs are lost. So over time, once this happens every replication, it just moves farther and farther down. So if that was the actual DNA of the chromosomes, you'd be losing important information, and the cell would stop working pretty quick. So this is a little video, just kind of explains a little bit better than me.
The ends of linear chromosomes pose unique problems during DNA replication. Because DNA polymerases can only elongate from a free 3' hydroxyl group, the replication machinery builds the lagging strand by a backstitching mechanism. RNA primers provide 3' hydroxyl groups at regular intervals along the lagging strand template. Whereas the leading strand elongates continuously in the 5' to 3' direction all the way to the end of the template, the lagging strand stops short of the end. Even if a final RNA primer were built at the very end of the chromosome, the lagging strand still would not be complete. The final primer would provide a 3' hydroxyl group to synthesize DNA, but the primers would later need to be removed. The 3' hydroxyl groups on adjacent DNA fragments provide starting places for replacing the RNA with DNA. However, at the end of the chromosome, there is no 3' hydroxyl group available to prime DNA synthesis. Because of this inability to replicate the ends, chromosomes would progressively shorten during each replication cycle.
So you guys kind of get what's going on there, how it's lost. Alright.
What are you trying to get to?
The next slide, but I thought I was interacting with the video instead of the.
Let's go to arrow, the arrow. Arrow at the top. Okay, yeah,
This is the right slide. Alrighty. So what happens when telomeres get too short? Well, if they were allowed to keep going, like I said before, it would end up destroying the chromosomal DNA. So then every cell after that would be damaged, or would have damaged DNA, and would probably just turn into a tumor. You'd have cancer. But cells have mechanisms to gauge the length of the telomeres. So when they get too short, they enter a stage called cell senescence, and they cease to replicate completely, unless the DNA gets damaged further and then gets reactivated. But talk about that a little bit later. So, like I was saying before, when you get older and you get a cut, all the cells around it is senesced, and most of them, so it takes a long time for it to heal. Not as much replication going on. This little thing was a chromosome at a barber shop. It says, "Don't cut it too short. I don't want to look old." So this is kind of like what's going on with the tumors. You've got a young, or just a cell with normal telomeres, and you've got a break in the tissue, something that happens. So it just starts replicating new tissue cells for the skin or wherever the damage occurred. And as each one's added, the telomeres get a little bit shorter. And then over time, it goes down, the cell gets old, and it's no longer able to replicate the cells. That's what you got here is this aged tissue, if you can read that. Just missing a bunch of pieces here and there. Or if there's a mutation that happens before it enters the senescent state, which is what's going on here, then it can get stuck. It can get stuck just replicating cells over and over, but quicker than what's happening here, you get a tumor. What's going on? Big old bunch of tissue cells with damaged DNA. So when does it usually occur? Well, we're all born with about 10,000 base pairs, depending if you're a girl or a guy, or what race, or genetic background or whatnot. And that's all we have for most of our cells, except for the reproductive cells can repair their telomeres. Each replication, you lose 50 to 200 base pairs. So I couldn't find a dead number, like when cells start to senesce for humans on average, but ballpark around 45 to 60 years of age. So what factors can affect shortening? Diet, both good and bad. If you have a bad diet, of course, it can affect it negatively. If you have a good diet, it'll affect it positively. Smoking can shorten telomeres just because it's damaging tissue cells. Your body's having to make more, shortening telomeres further. Same kind of, for UV damage, the UV rays hit the DNA and then get DNA damage, which can, or even just cellular damage, which causes the cell to need to replicate or enter a senescent state if the DNA is too damaged. Alcohol can also damage telomere. Depression and psychological stress. There's a study done that showed that these actually have a pretty heavy impact on your telomere length. They can shorten it if you're always stressed out.